All right, everyone, welcome back. Ready for another deep dive.
Always.
Today we're looking at, well, it's C. elegans, but not the typical kind. This time it's the blood-feeding kind.
Yeah, you don't usually think of those guys as bloodsuckers.
Right,
Is
it's
it?
kind of a surprise.
Yeah.
But this is actually huge, especially when we're talking about fighting those parasitic worms. The ones that cause so much trouble around the world.
Absolutely. Like it's hard to overstate the impact of parasitic worms.
Yeah, for sure.
So to be clear, we're talking about uh, C. elegans, tiny transparent worms, about a millimeter long.
Right, the usual lab workhorse.
Exactly. They're normally found in the soil, happily munching on bacteria.
So not exactly, the stuff of nightmares.
Not usually, no.
But this research, this is really turning things upside down.
Yeah, it's a game changer.
So what made scientists think to turn these little guys into, well, tiny vampires?
Well, the thing is, parasitic worms, like hookworms, for example,
Yeah, those are nasty.
they are. And they're a real challenge to study. I mean, their life cycles are complex. They need a host to survive.
It makes vaccine development a real headache.
It really does.
So I guess scientists wanted something, a bit easier to work with.
Yeah, a simpler model, something they could really manipulate to study parasitic worm behavior, especially their blood feeding, the hematophagy.
Okay, so C. elegans is the stand-in, the understudy for the villain of the story.
You could say that.
So how do you even go about making a worm that usually eats bacteria start feeding on blood?
Right. It's not like you just offer them a little dish of blood and hope for the best.
You'd think it would be that simple, right?
Well, actually, it all started with donated human blood.
Okay.
The researchers carefully isolated the red blood cells from the blood.
All right.
And then they did something pretty cool. They labeled those red blood cells with this fluorescent marker.
A fluorescent marker. Okay,
Yeah.
now I'm lost. Break that down for me.
Sure, a fluorescent marker is basically like a dye that glows under certain kinds of light.
Yeah.
By labeling the red blood cells, the researchers could track them, see what happens when the worms encounter them.
Ah, so it's like a little tracking device.
Exactly.
So what did they see? Did the C. elegans just slurp those blood cells right up like a tiny little milkshake?
It's actually a lot more complicated than that.
Yeah.
They consume those red blood cells in a 5-step process. Can you believe it?
5 steps
Yeah.
for something that tiny.
I know, it's wild.
Walk me through it.
So first they have to capture the red blood cell, which is, a red blood cell is much larger than their mouth opening.
That's got to be a challenge.
You would think.
Right.
It's kind of like trying to swallow, I don't know, a watermelon whole.
Okay, so how do they do it?
They use their pharynx, basically their throat, to rupture the blood cell.
So it's more of a bursting situation than a graceful sip.
Pretty much, yeah.
Sounds.
And then they draw the contents of the burst blood cell into their intestines.
So they're not exactly the tidiest eaters.
No, not really. The whole process is actually pretty inefficient. There's some spillage.
Sounds like a microscopic bloodbath. But can they actually survive on this? I mean, do they thrive or just barely get by?
What's amazing is that the research showed that C. elegans, they can actually thrive on a diet of red blood cells alone.
Wow, full on vampire mode.
Yeah.
So does that mean they have some secret I don't know, blood digesting superpowers?
Well, they did find that C. elegans has certain enzymes, like aspartic protease 4
Okay.
and GST, which are, very similar to ones found in parasitic worms.
Hold on, back up. Enzymes? What are those again? And why are they important here?
Right, so enzymes are basically proteins that help break down different substances.
Okay.
In this case, these aspartic protease 4 and GST, they're crucial because they help the worms digest hemoglobin, which is the protein in red blood cells that carries oxygen.
Ah, so that's how they're getting their nutrients from the blood.
Right, and they also help to detoxify the harmful byproducts of consuming blood.
So no enzymes, no blood-sucking diet.
Pretty much. And this is where things get really interesting because it ties into how we fight those parasitic worms we talked about earlier.
Okay, so tell me more.
Well, the thing is,
So these enzymes, the aspartic protease and the GST,
yeah,
they help C. elegans digest blood, but they're also similar to what you find in hookworms, right?
exactly. That's the key point here.
Okay, so the hookworms, those are the bad guys, the ones that infect people.
Right. And scientists believe that these enzymes in hookworms, they could be the key to developing vaccines that actually work.
So by studying these these vampire C. elegans, we can learn how to fight the real deal.
Yeah, that's the idea. It's like having this simpler model, a controlled environment where we can, test out different strategies without all the complications of working with the actual parasite.
So it's like a training ground, a way to practice our moves.
Exactly.
Yeah.
And remember how we were talking about how hard it is to study parasitic worms because they need a host to survive?
Yeah, that makes things so complicated.
Well, C. elegans, they don't have that limitation. We can grow them in the lab without needing to, infect any animals.
That makes things a lot faster, a lot more efficient.
It does, and it opens up the possibility of testing way more potential vaccines.
So this could really speed up the whole process of developing new treatments.
Absolutely, yeah. And there's more. It's not just about those two enzymes we talked about.
Really? What else is there?
Well, they're also looking at, other molecules that are involved in this whole blood feeding process,
Okay.
like lipases, for instance.
Lipases, what are those?
They're enzymes that break down fats.
Fats in blood.
Yeah. Blood has fats too.
Huh. Learn something new every day.
And they're also looking at NUC1, which is a nuclease, helps to break down DNA.
Wait, DNA, what's that got to do with blood?
Well, some parasitic worms, they actually feed on white blood cells. And those are, full of DNA.
So it's not just a red blood cell thing. These parasites they're really going all out.
They're very adaptable.
Yeah.
And then there's CRT1.
Okay.
This one's a protein involved in all sorts of cellular processes, like the way cells respond to stress.
Okay, so by targeting all of these different molecules, I guess that gives scientists more options for disrupting the parasite's blood feeding.
Right. It's about finding multiple ways to attack the problem.
This is fascinating. We've gone from talking about these tiny little worms to potentially, groundbreaking strategies for fighting these diseases that affect so many people.
It really is amazing how something so small can, have such a big impact.
It makes you wonder what other secrets are hidden in these creatures, just waiting for us to discover them.
Yeah, it's a reminder that there's still so much we don't know about the natural world.
Well, this deep dive has certainly taken us down some unexpected paths.
It has, but it's been a fascinating journey, wouldn't you say?
Absolutely. We've looked at the science, the potential, the mysteries surrounding these blood-feeding C. elegans.
And it all points to, the importance of staying curious, asking questions, exploring the world around us.
I don't think I'll ever look at a tiny worm the same way again.
Me neither. And who knows what other incredible discoveries are waiting to be made just beneath the surface.
So we've been talking about this research, C. elegans feeding on blood.
Yeah, it's pretty wild.
And how it could change the way we fight those parasitic worms.
It really could. I mean, imagine the impact this could have. So many people suffer from parasitic infections.
It's amazing how these tiny little creatures can open up these huge possibilities.
It really speaks to the power of scientific curiosity and just following the evidence wherever it leads.
And this research, it also it makes you think about evolution,
Oh, yeah. In what way?
Well, like the blood feeding thing, the haematophagy. It makes you wonder, like, where did that come from? Did these worms have a blood-sucking ancestor somewhere way back when?
It's a fascinating question. It's like, did we just kind of awaken this hidden potential in them? Or is there like an ancient lineage of blood-feeding C. elegans out there somewhere?
Like a secret society of vampire worms.
Right. And this research, it could be a starting point for, a whole new line of investigation looking into the evolutionary history of these guys, how they've adapted to different food sources.
It's like we've uncovered this hidden talent, this secret skill they had all along.
Absolutely. It just goes to show how adaptable life really is, how even these tiny creatures can hold such amazing secrets.
It's mind-blowing to think that these worms could be a window into millions of years of evolution.
It is, and it just makes you realize how much we still don't know about the natural world.
Well, I think it's safe to say that this deep dive has been quite a journey.
It really has, full of surprises.
Yeah. We've explored the science, the potential, the mysteries, all thanks to these blood-feeding C. elegans.
And it all highlights the importance of staying curious, of never losing that sense of wonder about the world around us.
I don't think I'll ever look at a a tiny worm the same way again.
Me neither. And who knows what other incredible discoveries are out there just waiting to be made.
That's right. There's a whole universe of possibilities out there just waiting for us to explore them.
So keep exploring, keep asking questions, and who knows, maybe you'll be the one to make the next big discovery.
Well, folks, that wraps up our deep dive into the world of blood-feeding C. elegans. It's been a real pleasure having you with us.
Thanks for joining us.
And until next time, keep those minds curious and stay hungry for knowledge.